Good afternoon, students. Today I am back with my next class on biological classification. Till last class, we learned about classification by Linnaeus, and he divided the whole living world into two kingdoms, Plantae and Animalia. But there he ignored following points, that is, differentiation between eukaryotes and prokaryotes. So, what is that? Like in Plantae, there were fungi, there were unicellular bacteria, algae, as well as higher plants, as well as higher plants. They all were inside kingdom Plantae. So we can say that there was no differentiation between eukaryotes and prokaryotes. There were also no differentiation between unicellular and multicellular. We can see here that unicellular bacteria, multicellular fungi, obviously multicellular higher plants, all are present together. Differentiation between photosynthetic and non-photosynthetic were also not there. Since we know that bacteria can also be photosynthetic, right? Like cyanobacteria. Bacteria which is photosynthetic is known as cyanobacteria. So it is also present in the same Plantae kingdom where non-photosynthetic fungi are also present. So there are 3 points which were ignored while doing Linnaeus's classification. That is differentiation between eukaryotes, prokaryotes, differentiation between unicellular, multicellular, and also differentiation between photosynthetic and non-photosynthetic organisms. So, from Linnaeus's classification evolved another type of classification which is known as Whitaker's five kingdom classification. Which is known as Whitaker's five kingdom classification. So now we are going to study Whitaker's five kingdom classification. So what did R.H. Whitaker proposed? He proposed that there should be a five kingdom classification where the whole living world is divided into Monera, Protista, fungi, Plantae, and Animalia, where it is divided into five kingdoms: Monera, Protista, Fungi, Plantae, Animalia. Particularly in this lecture, in this video, we are considered about Monera and Protista. In my later videos, I will go through in details about fungi, Plantae, and Animalia. Okay, so now let us understand the basic differences between these five kingdoms. So what is the first criteria? That is the cell type. In case of Monera, it is prokaryotic. In case of Monera, it is prokaryotic. In case of Protista, this is eukaryotic, 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 eukaryotic. So we can remember, other than Monera, all other four are eukaryotic. Okay. So I think that this kind of a chart would help you. So I have given it in this chart form. I think it would be easy to remember if you see in the chart form. So now comes cell wall. The next criteria is cell wall. In case of Monera, there is cell wall, but that is non-cellulosic. That is non-cellulosic. What it is formed of? Polysaccharide amino acids. In case of Protista, cell wall is not mandatory but present in some. In case of fungi, cell wall is present but with chitin. In case of Plantae, we know cell wall is cellulosic. And in case of Animalia, it is absent. So now our next criteria is nuclear membrane. Obviously, in prokaryote, since it is a prokaryote, nuclear membrane is absent. Prokaryotes never have a well-defined nuclear membrane. Prokaryotes can never have a properly distinguished nuclear membrane. So next, all the rest of the four are eukaryotic. So membrane is present, present. So we have to remember that in prokaryotes there can be no well distinguished nuclear membrane. Now Monera, unicellular, multicellular, loose tissue. Protista can be unicellular as well as multicellular, but cellular. There is no uh, formation of organ, tissue, this kind of higher organisms in Protista. These two are cellular. In fungi, it is multicellular and loose tissue can be formed. In case of plantae, we know that there can be tissues and organs. And in case of animals, we know tissue, organ system. Okay, like what is the organ system? Like nervous system, digestive system. So we can also have organ system. Now these are predominantly autotrophic Monera, that is bacteria. Bacteria can be different types of autotrophic like chemosynthetic, photosynthetic or heterotrophic. So it can be, autotrophism can be of multiple types like chemosynthetic, photosynthetic, saprophytic, parasitic. In case of Monera, the autotrophism can be of several types, that is chemosynthetic, photosynthetic, saprophytic, parasitic. In case of Protista, autotrophism is mostly of two types, photosynthetic, heterotrophic. In case of fungi, they are mostly heterotrophic, and within heterotrophic, they are generally of two types, saprophytic and parasitic. Plantae is autotrophic, we all know, and it's mostly photosynthetic. Okay, so now comes Animalia. Animalia is heterotrophic and it can be holozoic or saprophytic. So these are the different types of kingdoms with their very specific features. Now we are going to go in details about our first kingdom, that is Monera. So comes our first kingdom, Kingdom Monera. Monera kingdom has a sole kind of microorganism which is bacteria. The sole member of Monera is bacteria, and it is the most abundant. It is the most abundant microorganism. Till now, it is the most abundant microorganism. Now, bacteria are generally grouped into four categories based on their shape. One is these round-shaped bacteria, which are known as cocci, rod-shaped bacteria, which is bacilli, filamentous bacteria, known as spirilla, and this comma-shaped bacteria are known as vibrio. So bacteria are generally grouped under four categories. Okay, they are grouped under four categories. One is round-shaped cocci, two is rod-shaped bacilli, third is spiral or filamentous spirilla, and fourth is comma-shaped vibrio. Now points to remember. Now, there are few specific points to remember about bacteria. I think only these points are enough to learn from that big paragraph in NCERT. That is, they mostly can synthesize their own food from inorganic substrates. They can be as well as photosynthetic, and they depend on other organisms for dead organic matter. So they can gather their food in three different ways. That is, they can synthesize their own food from inorganic substrate, they can synthesize their own food by photosynthesis, and also they can depend on other organisms or dead organic matter for food. Okay, so these are the three important points that we had to remember about bacteria. And next comes two divisions, that is archaebacteria and eubacteria. There are primarily two divisions in bacteria, archaebacteria and eubacteria. So what is archaebacteria? It has a very unique feature of staying in harsh conditions. We can see extreme salty condition, extreme hot condition where bacteria will be there. Extreme salty conditions are known as halophiles. Bacteria remaining alive in extreme salty conditions are known as halophiles. Bacteria remaining alive in extreme hot conditions, that is in hot springs, are called as thermoacidophiles. They are known as thermoacidophiles. And bacteria in marshy area, wait one second, let me delete this. Bacteria in marshy area are known as methanogens. Archaebacteria has certain differences from the other bacteria. What? That is in their cell wall structure. Because their cell wall structure is so that it helps them to sustain all those harsh conditions. So they differ from the other bacteria in cell wall structure. And one very interesting thing about methanogens is that methanogens are present in gut of cows and buffaloes. So they are basically responsible for production of methane from cow dung. Nowadays we know, no, that from cow dung we are getting biogas. It's in such a height. So because of this methanogenic bacteria which is present in the gut of cows and buffaloes, we are able to produce methane from the cow dung. So archaebacteria is done. Now we will be moving to eubacteria. So this is a particular example of eubacteria. This is a basically blue-green algae. We also known as cyanobacteria. Cyanobacteria. So this is a part of eubacteria. Okay. Although we know it as blue-green algae sometimes, but it is under bacteria and it is under eubacteria. And we have to remember that this is a photosynthetic organism. This is a photosynthetic organism. So what are eubacteria? These are the true bacteria. They have rigid cell wall. They are motile if they have flagellum. And in case of cyanobacteria, they have chlorophyll A, just like in the higher plants. Okay. Cyanobacteria are unicellular, colonial or filamentous. And the colonies are generally surrounded by mucilaginous sheath. This blue colored thing you can see. This is the mucilaginous sheath. So colonies are surrounded by mucilaginous sheath. Some eubacteria can fix atmospheric nitrogen in cells called heterocyst. Here there is heterocyst. These are the heterocysts where atmospheric nitrogen can be fixed. They can be chemosynthetic, that is they can use other inorganic substrates for making their food. What are those? Nitrite, nitrate, ammonia. for and use that energy for ATP production and use the energy for ATP production. So they are helpful in recycling nitrogen, phosphorus, iron and sulfur. Therefore, they are helpful in recycling nitrogen, phosphorus, iron and sulfur. Now comes few important characteristics like heterotrophic bacteria can be very important for the ecosystem because they are very important decomposers. Not only ecosystem, actually for our food also, because heterotrophic bacteria, which are important decomposers, help us in making card, antibiotics and also fixing nitrogen. Some bacteria can also be pathogenous, like cholera causing, typhoid causing and tetanus causing bacteria. What is the typhoid causing bacteria? It's Salmonella typhi. Typhoid causing bacteria is Salmonella typhi. So few bacteria are pathogens. They reproduce mainly by fission, which I have shown here, like a dividing bacterium. This is a dividing bacteria. It will be clipped off from here. Now this will be one bacteria. This will be another bacterium. So one bacteria can divide and form two bacteria. So they generally reproduce by fission, but sometimes sexual reproduction is also seen with a very primitive DNA transfer method. Another organism present in this kingdom is Mycoplasma. Mycoplasma lacks cell wall. They are till now identified smallest living cells. They lack cell wall. They are till now identified smallest living cells. They can survive without oxygen. They are pathogenic to animals and plants. Okay. So with Mycoplasma, classification of Monera or characterization of Monera is done. Our next part or topic will be Protista.